Hi, my name is Connor O'Brien, and I'm a senior biological sciences major at Clemson. Over the past year, I've conducted research on crustacean mitochondrial genomics with Dr. Beza as part of the Clemson Creative Inquiry program. If we begin by looking at crustaceans belonging to the infraorder Brachyura, known as true crabs, we find the family Ocypodidae. This family consists of semi-terrestrial crabs found across the world in both tropical and temperate beaches. Within this family, there are three subfamilies: Eucinidae, that contains mangrove crabs; Eucinae, or fiddler crabs; and Ocypodidae, or ghost crabs. Ghost crabs are most commonly known for being one of the fastest extant crustaceans on land. Some have been observed running at top speeds of 2 to 4 meters a second, and they travel up to 300 meters a night while foraging. Other features include elongated eye stalks, asymmetrical chilopads, stridulating ridges, and a dull color to blend in with the sandy substrate. The focus of my talk today will be on the Atlantic ghost crab, otherwise known as Ocypode quadrata. This is a medium-sized semi-terrestrial crab inhabiting sandy beaches ranging from Rhode Island in the United States to Rio Grande do Sul in Brazil. If we take a look at their diet, we find that they are nocturnal scavengers, eating organic dead material and small live prey, including mole crabs and coquina clams. When they are not searching for prey, they are typically found within burrows in which they remain for the majority of the day. They are an important ecological component in sandy beach environments and have often been used as a bioindicator of human impact on sandy beaches. Prior to this study, the ecology and physiology of ghost crabs have been well characterized. Specifically, many have looked into the physiology associated with its running behavior. Despite this, there are currently limited genetic and genomic resources for Ocypode quadrata. This makes it difficult to draw connections between the heavily studied behavior of the crab and any associated genetic underpinnings. In this study, we assembled and characterized the mitochondrial genome of Ocypode quadrata to provide novel information about this important semi-terrestrial inhabitant of sandy beaches worldwide. We also wanted to confirm phylogenetic relationships within the Ocypodidae family. This would allow us to compare to results found in a recent paper on Manuka monax, also in the Ocypodidae family, done by another member in the Beza lab. To begin, specimens were collected and the mitochondrial DNA was extracted from tissue samples. These are sequenced using Illumina next-generation sequencing and the program Novoplasty to de novo assemble the genome. This process was conducted as part of the creative inquiry, but not by myself. For initial annotation, the assembled genome was uploaded to the Mitos and Mitos2 web servers using the invertebrate genetic code. This predicted the locations of genes, but the full curation, including start and stop codon locations, was done by me using the web server Xpasi and the MegaX software. After producing an accurate curation, I used Genome VX to visually depict the mitochondrial genome. Using several softwares, I was able to analyze several features of the mitogenome. This included the nucleotide composition of the whole mitogenome, the codon usage within the protein coding genes, the selective constraints within the protein coding genes, and the secondary structure prediction and depiction of the tRNA genes. I also analyzed the control region to predict potential secondary structures and look for evidence of organization, the presence of microsatellites, or tandem repeats. Also, a phylogenetic analysis was run for Ocypodidae. As seen in the provided genome map, the complete mitochondrial genome was found to be 15,547 base pairs in length. This includes 13 protein-coding genes, or PCGs, 22 transfer RNA genes, and 2 ribosomal RNA genes, rRNL and rRNS, corresponding to the 12S ribosomal RNA and 16S ribosomal RNA subunits respectively. The L strand or light strand contained 9 of the PCGs and 14 tRNA genes. This can be seen in the graphic as the outwardly facing genes. The H strand or heavy strand contained the remaining 4 PCGs, 8 tRNA genes, and the 2 rRNA genes, represented on the graphic by the inwardly facing genes. The mitochondrial genome was found to be compact with little intergenic space or overlap between genes. The 685 base pair intergenic space between the rRNS and tRNA genes was presumed to be the D loop or control region, involved in the initiation of DNA replication. This was of similar length and position to that seen in its congeners. Included here is a summary of the findings from the gene order curation. The gene order of Ocypode quadrata, as seen in the middle panel, was identical to that seen in other Ocypode Ocypode species, as well as most other brachyrhine crabs. Interestingly, the data previously obtained from Manuka monax shows a slightly different gene order. A former student in our laboratory observed a switch in the position of the tRNI and tRNQ genes that is not seen in any other Ocypodidae species. For Ocypode quadrata, the position of the tRNI and tRNQ genes were consistent with the species belonging to the Ocypodidae family. Another notable observation for the gene order was the location of the tRNE, tRNF, and tRNH genes. Ocypode quadrata also showed the translocation of the tRNF gene between the tRNE and tRNH genes that has been observed in other brachyrhine crabs. This contrasts with other decapod infraorders, which have been observed to contain this tRNF gene between the NAD4 and NAD5 genes. If we look at the nucleotide composition, we can see that the genome showed an AT bias of 70.02%, which is within the known range of 65 to 78% for other semi-terrestrial crabs, and within the range of that seen for other Ocypode species with data available. For the codon frequency found within PCGs, we can see similar trends to that seen with the nucleotide composition due to the strong AT bias. The most frequently used codons were those consisting of thymine and adenine, with the exception of leucine. The least frequently used codons were primarily those consisting of cytosine and guanine, with the exception of cysteine. It should also be noted that the COX3 and COB genes ended with an incomplete T-stop codon. These have been observed in its congeners and are speculated to be completed post-transcriptionally via polyadenylation of the mRNA strand. I'm going to elaborate on the methods behind a few analyses in particular in order to fully understand our findings. For the selective constraint analysis of the protein-coding genes, we used the Ka-Ks calculator 2.0, using a pairwise comparison with Ocypode quadrata, a closely related species. This program calculated Ka, Ks, and omega. Ka represents the frequency of non-synonymous mutations, while Ks represents the frequency of synonymous mutations. Omega represents the ratio between Ka and Ks. Thus, omega ratios of less than 1, equal to 1, and greater than 1 indicate genes undergoing purifying, neutral, and diversifying selection, respectively. For the selective constraints on the PCGs, we saw that all of the genes were undergoing purifying selection. The Ka-Ks for ATP8 was an order of magnitude greater than the COX1, COX2, COX3, and ATP6 genes, and at least 2 times greater than the remaining genes. This indicates strong purifying selection for the COX1, 2, 3, and ATP6 genes, and the weakest purifying selection for the ATP8 gene. This selection pressure analysis has not been done on any other species in the genus. However, this has been done in Manuka monax within the family Ocypodidae, which also showed the strongest purifying selection in the COX genes and weakest for ATP8. The general trend of purifying selection has remained consistent for the PCGs of several decapod crustaceans. If we now look at the tRNA genes, it was found that they all ranged from 64 to 70 base pairs in length. All the tRNAs showed a conventional cloverleaf structure except for tRNS1. This gene was missing a D arm, and a similar phenomenon has been observed for this gene in all other brachyrhine crabs with secondary depictions available. Lastly, the phylogenetic position of Ocypode quadrata, as well as other representatives of the family Ocypodidae, was studied. A phylogenetic analysis was performed using the MitoFast program. The genomes of 3 species from the Ocypodidae subfamily and 9 from the Gelaciminae subfamily were used as in-groups for the analysis. 2 species belonging to the family Xenograpsidae, 2 belonging to the family Gecarcinidae, and 1 species belonging to the family Grapsidae were used as outgroups during the analysis. This MitoFast program was able to perform a maximum likelihood phylogenetic analysis using the software IQ tree, with bootstrapped iterations of the observed data 1,000 times. These results fully supported the monophyletic status of the family Ocypodidae and the monophyletic status of the 2 subfamilies, Ocypodidae and Gelaciminae. Within Gelaciminae, these results were in agreement with the findings of a recent mitogenomic study by another CI student. This supports results from studies focusing on nuclear genomes, so this again establishes the utility of mitochondrial DNA for use in phylogenetic studies. This was the first study to assemble and characterize in detail the full mitochondrial genome of Ocypode quadrata, an important bioindicator species in sandy beach habitats in the Gulf of Mexico and northeastern Atlantic coast. This study provides a genomic resource to improve our understanding of the adaptation of semi-terrestrial species to sandy ecosystems, as well as the acclimatization of crustaceans to global climate change. It is hopeful that understanding species such as Ocypode quadrata can contribute to efforts to restore and/or maintain beach ecosystems.
Thanks for listening.